using machine learning, separate those that look more like tracks or look more like showers, like an electron, for example. So it sounds very different to what I do now, but at some point, as I said, I wanted to do something more impactful, and I found my second postdoctoral position in the Department of Radiology, where I am now. And for a few years, I was also part of CRUK and NCITA, based on cancer research. It's a network, a consortium of different centers in the UK working on developing these tools and these pipelines for cancer research. So I've done a few different things. I've worked on building databases or repositories for data sharing. I'm working on some federated swarm learning as well. And the part that is probably most interesting for today is the work that I've done with people in my department, in my group, and PhD students on deep learning for tumor detection and segmentation, and machine learning for tumor classification and prediction. So I'm going to be focusing on those two use cases for today. And yes, it is, you might be thinking that those images at the beginning look very different to what we see in radiological images, which is what I work on now. But I think it's during our training as physicists in particular, or as data scientists or mathematicians, I think we developed this very abstract thinking, and we can very easily translate the pattern recognition or the separating these tracks of these particles into any other different type of images. I have worked in the past a little bit on X-rays, ultrasounds, just in some applications, but mostly CT, with a little bit of PET as well, and now starting to develop some things in MRI. But CT is mostly what I work on because, as we all know in the UK, that's the standard of care imaging. When a patient is suspected cancer, they get a CT for diagnosis, so it's what we have the most images for, and what is most useful to develop tools in the clinical setting, so they can be applied for any patient. And I do this work as part of the Radiogenomics and Quantitative Image Analysis group here in Cambridge. Here you have our picture of us, very smiley, because we were on a summer retreat somewhere sunnier than Cambridge. So we were all very happy. And you have also the link to our Twitter if you want to follow our research, and our website, which at the moment is under maintenance. So I promise you that we exist, even if the website at the moment is down. And we are a really interdisciplinary team, which I think is our main strength and something really enjoyable to work on these kind of problems. When you can share experiences and discussions with a group that contains physicists like myself, engineers, biomechanical engineers, mathematicians, computer scientists, and of course, the clinical experts, radiologists and oncologists, the most of them. This group was founded by our former PI, Professor Evi Sala, whose main expertise is ovarian cancer. Hence, a lot of work we've done is in ovarian cancer. And since a few years ago, I've been pushing, keep leading this group. And we are inside the hospital, so we are inside the Addenbrooke's Hospital of Cambridge University Hospitals, but we have very strong collaborations. We are like the pillar where the images are acquired, and we have these strong collaborations with other groups, particularly with the Department of Applied Mathematics and Theoretical Physics, in particular with the Cambridge Image Analysis group in this department, and also with CRUK Cambridge Institute as well. As I said, we work mostly on ovarian cancer these last few years, but it's not the only cancer we are looking at, which means dealing also with a lot of variability of how these cancers may look like. So my first study, it was actually on liver tumors, liver cancer, HCC, and then I've done also quite a bit of work on kidney tumors as well, RCC. But today we're going to focus on ovarian cancer. And this is a very metastatic, very aggressive cancer. It develops, it's a multi-site cancer, so it will appear in different parts of the body, but these are the two most common sites in which it will be present, which is called pelvic ovarian tumor or pelvic ovarian disease, and omental tumor, which is the omental wall. Now, let's start thinking about AI and quantitative image analysis. We work on this because we want these tools to be present in the clinical setting. We want this to be used for patients, for real patients. And it's AI is getting its position and is getting stronger and stronger into achieving this, into being present in the clinics. At present, there are already AI-based tools that are routinely used by physicians that support in their decision-making, reading reports, doing reconstruction. Some of the scanners already have a deep learning-based reconstruction algorithm that comes with the latest versions of the manufactured scanners. But we want to go further. So what we are thinking is the future. With these tools in the future, we want to enable personalized therapies. We want to extract as much information as possible from the patient, and we want to have automatized ways that can tell us, can make predictions about how the patient is going to respond to a treatment and can inform us on what would be the best pathway for that particular patient. And one thing in particular, for example, one example is we know that more heterogeneous tumors, or with more heterogeneous types of tissues within the tumor, we know this would respond worse to chemotherapy treatment. So this is one particular problem that one particular question we've seen in ovarian cancer that we are trying to understand better from the imaging point of view. Can we measure that heterogeneity? Can we know a priori how the response is going to be? And I work mostly on, or almost entirely on imaging, is what I really like. I'm a very visual person. I like looking at images. So this is the a schematic or a cartoon of what I call a life cycle of the image. So from the moment we have the patient in the scanner, we have an acquisition of some physical measurements. In the case of CT, we would have raw data called sinograms, which look like this, and we need to reconstruct those sinograms into something that we can understand, something that shows us the inside of the body where we want to find, for example, a tumor. So then, obviously, what we want at the end is to have a classification, for example, tumor type, or make a prediction if the patient will respond or not, or make a prognosis how long the patient is going to live. So these are all the steps, well, a bit simplified, but the different steps we need from the moment the patient is in the scanner and we acquire the images to the end, our end goal, which is extracting those answers or those predictions out of the images. And there are all these different stages, and for most of them, we can or we are developing AI tools for them, AI-based reconstruction, AI-based segmentation, also for feature extraction and modeling, using deep learning or using machine learning, also for data integration, data augmentation as well. I didn't put it for the data storage. I think I need to maybe update this slide a little bit, because you can also use transfer learning, or you can use other sort of things that would also impact the data sharing. And in addition, there are also other applications of AI that are relevant as well for radiological image analysis that can be NLP for reports as well, but I'm gonna focus only on imaging because that's what I work on. Of course, doing all of this has a lot of challenges, both from the data side and the clinical side as well, but one message I want to always give in these talks and with this particular slide is that each one of these challenges is an opportunity for us to develop a new method. So the methods that we have now, the novel technologies, methodologies we have, were responding to a previous challenge. So the challenges we have now are basically what needs to drive what will be the technologies of the future or the methodologies of the future. On the data side, the privacy preserving using of the images is, of course, something very important that we need to take into account, and we need to make sure that we preserve the privacy of those patients who are giving us the images to use. And for example, we can use federated learning, transfer learning, and things like this. a very straight away, very clear challenge that is still a very big challenge is that we are working with real-world datasets. So the datasets that we have come from different places, not only in terms of different people and different tumors, demographics, and so on, but also there's a lot of variability in terms of the protocol that's been used in different hospitals, the scanner manufacturer, the acquisition, and all of this makes extracting questions challenging because we don't have very big data sets. That's the last point. We never have enough data to develop our AI models. But so we need to be creative to solve this problem. We need to keep trying. We need to keep using this heterogeneity, dealing with the huge variability of tumors. And at the end of the day, we do this because we want something that is robust and is generalizable. We want these biomarkers to be robust and generalizable if we want to see them in the clinical setting, which has its own side or own set of challenges. We need to have a biological validation of the measurements, the biomarkers that we are extracting. We need to be able to interpret them. We need to be able to explain them if we are thinking about these models to be in the clinical, in any hospital being used. And we also need to take into account the public acceptance. And in the near future, we have to start thinking about ethics as well. If the model makes a mistake, who is liable for that mistake? And many more probably that I haven't put in here. But now let's go through those two examples I was talking about. So before we can extract measurements to make our machine learning models and classify or predict, we need to know precisely where the tumor is. So we need to do detection. We need to detect where the tumor is. This is the segmentation step. And now is the dramatic moment in which I drink some water and I ask the public if you can find the tumor. This is my pub quiz question. Can anybody guess where the tumor is in here, in this image? I can see. There are no clinicians on the room.
I think there might be. But no answers. Okay, go.
Yeah, so here we have a tumor that is in the right kidney that is presented visually on the left side. So in red is the two kidneys and in blue, that's the tumor. So that's, if I can go back, you could see something here, right? So this is something that maybe visually we would be able to understand or have a sense of what it is. But if we move to ovarian cancer, this is a lot more difficult. It's here. I don't know if you can see anything, if you can see something there. I've looked at a lot of these images and I can barely ever have a sense of where these tumors are. That was an example of pelvic ovarian disease, and this is even more difficult. This is omentum disease and is over here. So we are trying to find, we need to go towards AI and deep learning based tools to identify this automatically, because the processing based algorithms that we could use have limited applicability or will have limited accuracy or success in finding things like this, because there's no very clear pattern. It's very difficult to be able to do something like this just with processing tools. So it's with this type of very complex tumors, we need to go to a data-driven approach. Now, when we talk about detecting something, for example, the tumor, there are different ways in which what we mean about that detection. So the easiest way or the easiest detection would be I give you the image and you tell me there's a kidney or there's a kidney tumor. So you give me a label. That would be simply a classification. From this image, there are these two. In this image, there are these two labels. The next step would be what we call detection, which would be giving those labels, but also giving a spatial location, like a bounding box or a point, central point or something like this, something that gives us more, not just the label, but where around in the image those labels are. Those two are typically problems that you can solve with CNNs and also machine learning and deep learning combinations. In the case of segmentation, I'm talking about semantic segmentation. So this would be a problem of pixel by pixel classification. So here we are interested in every pixel in the image having a label. For example, kidney, kidney tumor, or background. So it's a more complex problem. It's more information because we want the precise delineation of these tumors. And this is a problem typically solved with DUNE. So I'm going to show you an example now in the case of ovarian cancer. So this is work done by Thomas Budenkotte, who was PhD student in the Department of Applied Maths and Theoretical Physics and working in collaboration with us. And this, I can see I need to update this slide. The paper has been already published. And this is some figures in that paper and in his PhD work. This shows you how this pelvic ovarian disease looks like in a few cases. These are manual segmentations. So you can see there's quite a lot of variability as well within this same cancer. It can be quite heterogeneous. It can be really large, like this case. And the example or the case with disease in the omentum or omentum disease, it would be even more complicated. If you look at these two cases in particular, these are very, the detail here is very granular. There are a lot of connected components, a lot of very, very large volumes as well. And this basically is something that takes our radiologists, some of these very complicated cases can take a couple of hours for them to do this manually. So it's not something that we can manually or that can be manually segmented for every patient. That's why we want to have an automated way of finding these tumors. And Thomas did this work with multi-class classification, so finding everything that was a tumor and then classifying that as pelvic ovarian disease, omentum, also lymph nodes. There are many different sites in which this disease can also appear. Even we have had some cases in which it can metastasize even to the lungs. And one thing that I want to point out in this slide is that when we are doing this, we are developing these tools and we are working on these problems in medical imaging, we also need to be conscious of what the ground truth is. We don't really know exactly the tumor. We rely on the experience and the expertise of the observers, the radiologists that are doing the manual segmentations for us. But there might be some variability between what two experienced readers or radiologists would segment, because there might be some room for discussion or some parts of that a person would consider tumor and another person wouldn't. So there's some variability that we have to consider. So when we are developing our metrics, we have to take into consideration that the ground truth can be more complex than simply ground truth. We have one answer for our image. So what we did in this case was trying, in the case of the omentum in particular, the agreement between two observers can vary quite a bit, and we found this is a subset of the cases that we use. And in this subset of particularly difficult cases, the agreement between two experienced observers was about 70%. So what we can aim to achieve is something similar to that number, so something close to what two different people would have as an agreement between them. And these measurements are in DICE score coefficient, which I will explain a little bit later. But how did we do this? So I said that we are using units, and I don't know if everybody's familiar here about units, so I have just a couple of slides very quickly to go through units. So they appeared in 2015 for segmentations of large biomedical images. They weren't radiological images. There were this type of images that are they were images of neuronal structures, and it was used on these data sets and as a windowing strategy. Tile strategy. Before that, the previously winners or state-of-the-art methodologies were based on sliding window CNNs, convolutional neural networks. The reason why units have worked so well for these very large volumes of biomedical images is that they combine a contracting path, which is basically a convolutional network. So this part where we are going down in the U shape, which is extracting abstract features in a lower and lower resolution as we are going down. Of course, if we lower the resolution a lot when we are here, we then need to undo that process, because at the end, remember, we want a pixel by pixel classification. So we need to give an answer for every pixel in the input image, which means we need to regain or resample our answer so that it will correspond to the same input size that we got from the original image. So we need to have an expansive path, which is similar to the contracting path in terms of applying convolutions. The difference is we will have, instead of max pooling operations, we will have upsampling or upconvolution operations. And one of the strengths of the unit and why it's drawn as a U is that not to lose completely, entirely the information that we are extracting through the different stages in the contracting path, we have residual connections there, these horizontal lines, meaning we concatenate that information. We use it back when we are doing the expansive path to avoid losing a lot of information when we are doing the upsampling part. Well, that was hopefully a short but informative introduction or summary about units. There are different ways you can use it. What Thomas did for this work on the ovarian cancer was using the so-called non-new unit or NN unit, which is a system to test different configurations, different architectures, different hyperparameter tunings, and so on into a configuration package. And he had to do this, or one of the ways in which one of the models he implemented was doing first a low resolution network, so downsampling the image and finding a region with a first stage unit, then cropping that part and training a second full resolution network or full resolution unit. I have a second pub quiz question you probably have seen already here. Can somebody tell me why we need two-stage networks, or why would it convenient to have something like this? The answer is the same, for the same reason as the original unit work was using tiles or sliding windows, which is we have very large volumes of data that we have to work on. Typically, we have 512 times 512 times of the order of hundreds of those 2D slices. So our volume would be typically 20 million voxels or more. And that means just one training example, one patient and one time point. But in normal average GPU memories, we can only fit about 1 million voxels. So we can't put the entire 3D volume at once in our GPU. So we have to develop, this is a challenge, we have to develop different solutions, like doing these different two-stage networks and doing sliding volumes or sliding windows and other things like this. We also need to think about the pre-processing and the post-processing. So that's also some steps that go into the pipeline that Thomas created. So pre-processing, you have a very heterogeneous data set. You probably want to resize it, so resample so that all the voxel sizes would be the same. And post-processing is also important. We realized of a few things when working on this problem. And some of the issues in terms of geometric, like dice score, that we were seeing, we saw that some of them would be easier to solve by a post-processing step rather than, for example, adding more layers into your unit architecture. Because there were some, for example, small bits that were picked up as a tumor that were not or were too small. So things that we could pick up very easily and discard as an artifact. And we also did something interesting. We ran a Turing test with a group of people with different expertise, so from medical students to very specialized in ovarian cancer experts. And we gave them different segmentation, some of them manual and some of them were automatic, and we asked them to see if they could differentiate between the two, and we asked them for comments. And one of the things that made them being able to differentiate the two were, for example, spikes or not very smooth contours around the delineations. Because, of course, if you do something manually, you're probably going to do it more smooth than the algorithm that can pick up things with a lot of detail, so it can end up having a lot of spikes. So those things are things that we can incorporate into a post-processing stage and make our accuracy at the end, our DICE score, better. The DICE, I keep talking about the DICE similarity coefficient. We have it here is a way to measure the overlap between prediction and ground truth. We have the definition there, and is widely used in medical imaging segmentation problems. It's also something that you probably want, and you definitely want to incorporate into your loss function, because if you base it just in the cross entropy, that would be looking at all the pixels, background and foreground. But the reality is that you are going to have a very imbalanced data set generally. So the number of pixels that you are going to have for foreground is going to be a very small fraction of the background pixel. So you want something else that will look particularly at the prediction and the ground truth only. So this is a typical thing to add the dice loss or the soft dice loss with different weights. One thing we are also interested in is not only giving this segmentation, but also can we quantify the uncertainty on this segmentation. There are different ways out there in which people are already thinking about this problem, like training with different random seeds. And what Thomas did was slightly different. In a problem, you might be, depending on your problem, you might be more interested in allowing false positives or false negatives. For example, if you are doing a screening or if you are thinking about a treatment, there might be different answers to your problem. And what he did was modify the loss function so that he could incorporate different scenarios in which he could allow more sensitivity as a trade-off, less specificity, and the other way around. So in a range of having a lot more sensitivity, less specificity, or more specificity, less sensitivity. And he trained all these different scenarios, the same architecture, the same algorithm, with different scenarios, and combined them together to give this kind of heat maps that you see here, in which obviously the places that are in the center of the tumor, or tumors that are easier, like this case is kidney, you can see that in the center the algorithms, you get 100% agreement between all of the different algorithms that you have trained, as opposed to around the edges is basically where you expect to have a lot more variability and to be more uncertain about the segmentations. So this was a good way of understand better the algorithm, increase our trust in what the algorithm was doing, and it has also other applications in the future. We were thinking about this would be a way in which we could create pseudo labels, for example, if we have unlabeled data sets, and to generate more training examples. And even we were exploring the idea of using this as a baseline segmentation for training other radiologists to do the segmentations that we need manually in the future. So it's a very interesting work that Thomas did there. I talk about UNES, and they are UNES, or based on UNES, are a lot of the algorithms out there that are getting the best accuracy, but they are not the only ones. And one thing I started a couple of years ago was a collaboration with my former colleagues in HEP, in particle physics, because I noticed that they were implementing a new way of, or they were using a different type of networks to deal with these images that are images in which, like this, you have a lot of space in this peak detector that is empty, there's no activity, and then you have a small fraction of the detector where all the activity is concentrated. So for this type of problem, something that can work with this sparsity of the image can be very advantageous, because you can basically restrict where the algorithm is looking at those parts where there is activity. So submanifold sparse convolution networks work like this. They have a slightly different version of what it would be a normal convolution, in which, remember, when you are applying your convolution, you are basically doing it with a window, for example, 3 by 3, meaning, say, whatever operation you're doing can be mean or max or any operation basically in your kernel, in your filter, but you would be doing this on not just on a pixel, but on a window. So you end up blurring your contours, blurring your image, making it less sparse. Instead, what we want is, in this sort of scenarios, is apply the same sort of convolution, but only keep as active pixels and positions that were active in the input. So basically do the same operation, but avoiding these blurring and blurring, like it gets more and more blurry every convolution. So we want to avoid that. We want to keep the sparsity of the original image. And this is good basically because it's advantageous because it gives us better performance and scalability. So you are avoiding to multiply a lot of zeros at the end. This is at the end of the day what you are doing. So you can by implementing this, you can have things that run faster and use less GPU memory. Of course, how do we apply this to CT images would be the question, because CT images by themselves, original images, are not naturally sparse, but in the case of CT, we can use the information that we have about Hansfield units to do a process of sparsification, meaning anything that is not within tissue ranges of Hansfield units, we can set it to 0. We know there's not going to be any tumor around here, or in our tumor cases, in bones or in anything that is in different tissue ranges. So we implement that as intermediate step of sparsification, and then we use the NVIDIA Minkowski engine, which is a submanifold sparse network, and we are getting very competitive results in terms of finding this. This is for kidney cases, so finding the kidneys and finding the tumors. And this is a project that has been the side project of everybody involved for a couple of years. So it's basically what we work on our free time. But the good news is that that archive there that has already a couple of years is soon going to be updated, because we have our final results coming very soon. So keep tuned, because it's going to be very exciting. Now, once we have our segmentation, we can extract some features and we can do some modeling. I work on what is called radiomics, which is a very widespread set of features that you can extract from these tumors, and they cover different categories. They can be measurements of shape, or they can be measurements of the pixel intensities building a histogram, these first order features, and any the typical histogram measurements that you can get, skewness, kurtosis, and so on. And then higher level features would be measurements of texture, which are based on the heraldic features that we know since the 70s, and they have uses in a lot of different applications. And they work by extracting or calculating an intermediate matrix that is looking at how many times pixels with two defined intensities are happening in the image. So it's looking at how many times you have combinations of pixels. And this can give us the measurements of heterogeneity that we want, as I said at the beginning, because we know they correlate with the response of the patient to chemotherapy treatment. And these are two examples, two recent papers in which we have looked at this for ovarian cancer, and we have found that these radiomic features can either predict by themselves in the second case, or can improve our prediction when combined from a baseline of having only clinical or CA125 or ctDNA, other blood or clinical measurements. So there is a promising to all these features, and we want to keep investigating them. Where are we at with radiomics? This is my personal view on them, and I like using this Gartner hype cycle for, I find that it can be applied to basically anything in life. So radiomic features is something that appeared between 2012 and 2015, 2014, and there was a peak of, in a couple of years, there were over 4,000 papers about it. But what we found is that it was very difficult to reproduce those papers or to make sense of two different papers, because either they were using very different data sets or they were using very different definitions of those features. So there's been a kind of a deep down there period, but we are getting out of it. There's a lot of initiatives like IPSI to standardize these measurements, and there's a lot of activity in this area, which is one I'm particularly interested in, understanding the robustness, the reproducibility of these measurements. These are two examples I work on in liver and in kidney CT, trying to understand what part or what fraction of those radiomic features actually can tell us things about the different tissue type or the different tumor type, or are we just, how much of what we are measuring is just differences in the acquisition, the reconstruction, even the variability between those two readers or two radiologists. In the case of the kidney, it was the same radiologist, but after a 6 months period. Of course, if after 6 month period, the same person will have very similar contours, but maybe 1-2 pixels up and down, we don't want this to affect massively our features and our prediction. So we want to avoid that. We want to find if some features get that behavior and discard them or correct for them. And of course, we do all of this because at the end, what we really want is have something that can be generalizable, something that we cannot afford having a person scan here in Cambridge and having an answer, and then the next day in London and having a different answer. We need to be able to give something that is generalizable, is robust, and we need to incorporate different data sets. And this is one example in that CRS prediction in which Leonardo was doing different internal and external cross-validation, and basically maximizing the generalizability or the difference between the two data sets. Because at the end of the day, we want to integrate it into the clinical setting. And this is also first attempt, a work that we've done a couple of years ago in collaboration with the whole NCITA network, and particularly ICR and with NVIDIA developers as well, in which we integrated into our repositories, our databases, this algorithm that Thomas created for automated segmentation of ovarian tumors, plus radiomics extraction and the prediction. And I can show you a video here very quickly. It's a demo of how it works. You have also the link to the paper there. So this would be a database where the radiologist can select a patient and they can open the images in a viewer like the viewers they are used to work with, and you probably are used to work with as well, in which we have incorporated our algorithm. So here there's a drop-down menu that is they're selecting segmentation, and now the algorithm created by Thomas and is running. I'm gonna pass this a bit fast because it takes a couple of minutes. In the meantime, this is what I see. The radiologists are back to that screen just waiting. But here I'm showing how we calculate the features and run the machine learning. And at the end, they get the answer that they want, which is which places have they found, the algorithm has found they are tumors, and they are colored in green in this case because it has, after the segmentation, radiomic features prediction, machine learning prediction finds that they respond or that they are predicted to respond those tumors. And yeah, maybe very quickly, the final bit I wanted to mention is I said also about biological validation, and this is something we have done in the past, basically with Maria, who was a PhD student with us until this year. She just graduated this summer. So there are two different, obviously, biological validation of these measurements. What we mean is we want to understand how they translate into a different tissue type or a different biology. To do that, we need to map it to a sample, a physical sample of those tumors. Two ways of doing it we have explored, either with biopsies, and in the case of the biopsy, what you have is a ultrasound image, and what we did was a live overlay of our analyzed CT diagnosis image on top of the ultrasound at the time of biopsy. So it will move with the ultrasound, so that we have previously analyzed this and found regions that have different values or different clusters of those radiomic features. So we can say, please take a sample from the green here and the red here, and they can, if possible, give us a way of connecting back to those radiomic features. The other way of doing it is with surgery. So when we create a 3D mold of the tumor and a 3D replica, so we can take this to the surgeon before the patient goes into surgery and the tumor is extracted. They can place it in those molds, and when it goes to pathology, they can cut in exactly the way we see in our CT image. So we can, we have orientation as well, so we can correlate and map from the tissue samples that will be processed afterwards in pathology back to the CT images. And finally, we need to keep the public on our side if we want to, if we want these tools to be used for real patients. And with this, I mean, we need to keep them informed of what we are doing. We need to keep explaining and having a very honest conversation about the things we are doing, how they work, and so on. So just my conclusions. I hope I have convinced you that AI-based methods are already proving to be very useful, and there's a lot of potential in using AI-based and machine learning-based methods to enable precision oncology and personalized treatments. But there's still a lot to do, and there's a very exciting research road ahead. We still have a lot of questions to work on, and I think interdisciplinary work is key to achieve all these impactful results, because we wouldn't be able to do, for example, the last example with the molds without the help of oncologists, radiologists, surgeons, and mathematicians, computer scientists, or data scientists like myself. So there's a lot of space to work on and a lot of really exciting problems to solve. And I hope you've liked it. Many thanks for listening, and happy to take any questions.
Thanks very much, Lorena. Of course, we did very impressive work and presentation. Thanks very much for sharing all of your exciting work with us. So, of course, if anyone has any question now, you can just put your hand or just open the microphone, and you can just make your comment, ask your question to Lorena. Or if you prefer, you can just type it in the chat. I can just read it for you. I have a first comment, if not, while people start asking. It's about validation of the models that you presented and that you said at the end of in your conclusion as well. So if I'm not wrong, you were using nested cross-validation, internal validation of models. How you feel about that? So are you planning to do any external validation? How difficult is to get the external validation on your models?
This one you're talking about for the prediction, well, all of our models really that I've presented today have external validation. So we don't work only with local data. And I think that's very important moving forward, that as I was saying, we cannot afford that something works here but doesn't work in a different hospital. We want these tools to be global. This is still, there's still like a long way to get there, because now I think people are more and more conscious that you need an external validation. And I think when you get your paper review, if you don't have it, they will probably go back to you and say, please find something. It, of course, has all the challenge of how can we share the data, and there are different ways of also dealing with this, like federated learning, if you cannot get the ethical approval and so on. So this is more and more in the space of we're working on. But we have to also start thinking about different demographics. Like, it's not just enough to compare maybe my hospital in Cambridge with the next hospital next door. We need to start going farther and farther with our measurements to start incorporating more acquisitions, more protocols, more demographics, a lot of different variability. But yeah, it's something we. So how I feel about it is we definitely need to do it, and we need to think about doing it more and beyond what we are doing. And we, in all our experiments, we've been lucky to have a lot of collaborations in which we have images from different centers. And some of these data sets, some of the time I've spent in this role is basically getting this big data set, these large data sets from different centers. But it's worth it, because at the end you have something that you can trust more, it's something more robust.
And it's more representative, is what you said. Thanks, Lorena. Is there any other question, comment?
I hope I didn't scare everybody with the slightly gory pictures with the 3D mold. It's very fitting for Halloween now.
If not, I have another question. So for your tool for automatic segmentation, it's about how clinicians felt about it. So were they really using it? Because, we were talking about the ethical considerations of this at the start of the talk. You said, no, maybe later, maybe in the future, but how do you really make the clinicians to trust your tool and so that they feel confident? Yeah.
So this work we did I have Cajal here. He was a medical student doing a PhD with us, and he spent a lot of time working on talking to patients about AI, and also doing this exercise that I said about the Turing test. So he has been investigating this, and his thesis is actually on our repository, so people can read about it. I think we need to start this conversation and giving them something that they can see, like being able to explain and say, this is the 90%, we are confident that this is a tumor, doing as much interpretability as we can. We do this also with our models. This paper with the cross-validation, for example, we were looking at clinical interpretable features only, for example. So things that we need to start having this conversation with them and showing them with an interpretable and explainability mindset, but also with, for example, doing the Turing test was something that they found fun to do, and so they can start believing, like if they can tell where there is an automated algorithm, but they still think it's okay, that makes them trust more the tools as well. So there's a lot of work to do, but there's a lot of fun things that we can do in this space. And in this public engagement events as well, we had some patients, and we had some stories that patients shared with us. For example, that we were asking them the question, would you trust AI to read your image, which is a question we need to start asking. And some people who have been in trials using AI said that definitely, that they were very happy AI was used for them because they got an answer much faster than other people. So in the future, I think this is, and radiologists generally, I think they are very happy to start incorporating these tools because it makes their lives easier if they can do things faster. It's not something that is going to replace radiologists, but I think it's something that is going to support them in the near future. If, for example, they can avoid having a second reader, but the second reader can be AI, that could be a way in which we could massively save time and money for the NHS, because it would reduce the expert time by half. So I think there's generally, I think there's a lot of like they're not against it. They want these tools to be implemented, but we have to be working with them. And the interdisciplinary work there is also a key part of how these things will end up being incorporated in the clinics.
Of That's very interesting, actually, because I think that you are in a field that you are really demonstrating that AI can, in some situations, surpass the human ability, and in identifying tumors, or And I was in the National Radiology meeting the last week, actually, and there were lots of interest and the ethical aspects on this, and lots of clinicians showing what you said, that they are starting to oh, that could be very helpful, and that could be saving time, probably just replacing one of the two readers, exactly what you say, maybe not both of them, but start to have some confidence on the tools, which I think that is very important and interesting in this field. Good. So there are any other comments? Don't think so. Okay, so a few minutes left, get to 4 o'clock. So let's finish the meeting. Thanks very much, Lorena. It has been fantastic to have you with us. I hope that you have lots of success in the future with your work, and let's stay in touch. Thanks very much. We will be meeting again on the machine learning seminars the next month on the 27th of November. I hope to have you all here again. Thanks very much for your attention, and have a lovely end of the day. Bye-bye. Thank you. Gracias, Lorena.